let's start today's class. So in the previous class, we had discussed about the various kinds of organisms according to their mode of excretion and what is the nitrogenous waste that they excrete. And we also saw the different excretory organs of different organisms. Now, did you people had any notes left to copy down, Aisha? Or was everything copied down?
I finished in the drawing of the human excretory system.
Which one? Which part?
Drawing of the human excretory system.
Drawing, okay. All right. So today we're going to talk about the human excretory system and the different parts it has and the function it performs. So right here on your screen, the picture that you see is the human excretory system, which contains a pair of kidneys. It contains a pair of ureters, one urinary bladder, and the urethral portion. Okay. So in humans, and also students, don't worry about the notes. Today I'm discussing the chapter through the NCERT directly so that we don't miss out any points, and I'll provide you with the notes. Okay. All right. So in humans, the excretory system, it consists of a pair of kidneys, one pair of ureters, a urinary bladder, and a urethra. So these are the four basic parts of our excretory system. Now talking about the kidney, so they are reddish brown bean-shaped structures. Where is it situated? So it is situated at the last thoracic and the third lumbar. So in between that situation. And it is like lumbar vertebra, and it is closer to the dorsal inner wall of the abdominal cavity. Dorsal inner wall means the, like for example, if this is the stomach portion in the front, so the back side, the kidneys are retroperitoneal in position, meaning it is situated towards the back side instead of being right in the front or in the middle. Now, each kidney of an adult human measures 10 to 12 centimeters in length, 5 to 7 centimeters in width, 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams. So the weight of each of the kidney is 120 to 170 grams. Now, right in the middle, do you see this part from where the ureters are coming out of the kidney? So that is like a notch. It's like an opening which is there, and that is called as a hilum. So through this hilum, ureters, the blood vessels, and the nerves enter inside the kidney. Okay, so it's like a notch-like structure. For example, if this is the kidney, so there will be a notch like this, and this will be called as the hilum. And through here, the ureter would arise, or ureter would come into it, and then also the different blood vessels and the nerves. Now, inner to the hilum is a broad funnel-shaped structure, which we call as the renal pelvis. If you see in this diagram. So, right inside the, like when you move inside the hilum part, right? So, right in the middle, this portion, this wide funnel-shaped portion, this portion is called as the renal pelvis. See, anything that is related to the kidneys, we call it as renal. Okay. So, this portion is the pelvis, renal pelvis, and then the projections out of the renal pelvis. That is another part which we see. So the projections is basically the funnel-shaped space, which was the renal pelvis that you people saw, and the projections were known as the calyces. So now you people are seeing the ultrastructure of the kidney. Now, the outer layer of the kidney, it is a tough capsule. Okay, it's like tough. And what does it do? It provides protection to the kidney. And inside the kidney, there are two zones that are present. The outer area inside the kidney, meaning the peripheral structure, will be called as the cortex, whereas the inner part will be called as the medulla. Now, the medulla, it is divided into a few conical masses. So, if you see over here, the conical masses are here, right? The cone-like structures that you see. These are the conical masses that are there. Okay. So, the inside the medulla portion, It is divided into the conical masses, which we call as the medullary pyramids, that projects into the calyx, or single, that is the calyx. Now the cortex, which was the peripheral portion, it's also going to be found in between those. Like for example, this is the situation. So this part is the renal pelvis, okay? And then here the conical stuff that you see is the medullary pyramid. And this portion, this one, this one, this one, in all of those areas, the cortex is going to be present. Is it clear, students? And that cortex is going to form a column, which is called as the renal column, or you can call it as the columns of Bertini. Okay. So this ultrastructure is clear. Now see, in this particular picture, you can see it very clearly, wherein you can see right in the middle, there is the renal pelvis, and these projections. These are the calyces that we were talking about, the renal calyces. And then on top of the renal calyces, you find these pyramid or this triangular structure, which is called as the medullary pyramid. And in between these spaces, all the light pink spaces that are there, that is the cortex. And the part of the cortex which was coming in between these calyces, we call it as what? It is the renal column, or you can call it as the columns of Bertini. This structure is also extremely important as far as the labeling, as far as the asking of like what are the functions of different parts and everything is concerned. Is it clear?
Yes, ma'am.
Okay. So now inside each kidney, how many nephrons are there? 1 million nephrons, which are the complex tubular structures, they are present inside each of the kidney. And the thing is that just like how cell is the fundamental unit of life, same way the nephrons, the functional unit of our excretory system is the nephron. Okay, so how many nephrons are present in each of the kidney? There are 1 million nephrons in each kidney. Now each nephron, it has two parts. If you see the picture, now see this picture right here. This is also again very important. They'll ask you to draw this in your like draw the structure of a nephron, draw the different parts of the nephron like that. So this picture that you see right in front of you, this is the picture of a nephron, wherein right in the middle, this part you see, this structure is the glomerulus, and the cup-shaped structure in which the glomerulus is present is the Bowman's capsule. Okay. Also arising from here are the different ducts like PCT, DCT, each of those functions you people will study and you people will understand it better. But for now, you understand that inside each of the kidney, there are millions of nephrons present, 1 million. Okay. And every nephron, it has two parts. The glomerulus, which is like sort of like a bundle of capillaries, a tuft of capillaries which was there, and associated with it, the renal tubules. Again, always remember the word renal is connected with the kidneys, always. What is the word hepatic or hepato connected with? Hepatic, hepato. No one. Hepatic related to the liver. Anything that is related to the liver that has the word hepato or hepatic associated. Okay, so now glomerulus is a tuft. Tuft means what, students? Tuft means like a bundle, like a bunch. So it's like a bunch of capillaries that is formed by the afferent arteriole, which is a fine branch of renal artery. So this renal artery that you see, further, when it is going to get branched, it's going to form the afferent arteriole. Now the blood from the glomerulus, see, later on, see, when, like, how would you study that which part is forming the glomerulus and which part is taking the blood away? So remember that blood comes into the glomerulus by the afferent arteriole and it leaves the glomerulus by the efferent arteriole. So it's like, how would you remember? Like when you have to call someone, you say idhar aao, isn't it? You say aao, right? So remember that the blood, when it has to come into the glomerulus, it will be A, like ow, blood ow. So that is afferent arteriole. You can remember it like this, or if you have a better way, you can remember it like that also. But this is my way of how I remember. It's like ow, so afferent. Okay, and then the rest that is left is efferent jaw. Okay, so the ow part is because of A, and that is afferent. And then this is whatever, jaw or bhago. Okay, understood? So the blood will be coming into the glomerulus by this afferent arteriole and it will be carried away by the efferent arteriole. Now the renal tubule, now the all like apart from this particular structure, all the other parts, all the other convoluted or like topsy-turvy things that you see, these are all tubules of the kidney. These are renal tubules, okay? All of them. So now the renal tubule begins with a cup-like structure that is called as the Bowman's capsule. This capsule-like structure that is there, it is called as the Bowman's capsule. Okay. Now, what happens is that the glomerulus along with the Bowman's capsule, meaning glomerulus plus Bowman's capsule is equal to renal corpuscle. Always remember, this is very important. So, renal corpuscle. So, that is the Malpighian body. You can also call it as Malpighian body or renal corpuscle. Now the tubule continues further to form a highly coiled network which is called as the proximal convoluted tubule. Now how would you remember this? See, once this cup-shaped structure, it has this extension and then it is going to be carried away, right? So it is going to be connected further. So proximal means near. Proximity is nearness of something, right? So the tubule which will be the nearest to the renal corpuscle will be the proximal convoluted tubule. Okay, so after that the tubule will be proximal convoluted tubule. Further, now see, further on, a hairpin shaped Henley's loop. Now, do you people see? Have you people seen those Judah pins that people put in their hair? It's like a U pin, the black colored, very thin. If you twist it, it gets twisted also, right? Those U pin shapes that are there, like we put it like this into the hair. So it's like that. Okay. So this hairpin like structure that you see, that is called as Henley's loop. Okay. And Henley's loop has two parts, descending limb and ascending limb. Now, descending means what? Top to bottom or bottom to top? What is descending? Like higher to lower, lower to higher.
Higher to lower.
Higher to. So, firstly, after the PCT, meaning after the proximal convoluted tubule is over, then it will go down. So, from top to bottom it is going. So, that is your descending limb. So, the first limb will be the descending and then it will come up and that will be ascending. So, it's like descending and then ascending like that. Okay, so the Henley's loop has two parts, descending limb and the ascending limb. So, hairpin shaped Henley's loop is the next part of the tubule which has a descending and an ascending limb. The ascending limb continues now further when this ascending limb goes to the next tubule and that tubule is distal convoluted tubule. Distal means far away. So, you can write this that proximal means near and distal means far away. Don't worry, all of these things will also be mentioned in your notes. But up until now, is everything clear, students? Any doubts? Anything you people want to ask?
No, ma'am.
Okay.
Ma'am, can you explain the Malpighian body again?
Malpighian body. See, if you look over here, we said that the nephron has two parts. It has glomerulus and then it has the renal tubules. Now this bundle-like structure that you see over here, this is the glomerulus. But this glomerulus is held in a structure which is a cup-shaped structure, and that is called as the Bowman's capsule. So when you collectively want to call them something together, what will you call it? You will call it as Malpighian body, or you will call it as renal corpuscle. Is it clear? It's like a collective name for both of them. It doesn't have any much bigger significance. It's just the name. So it's like many a times they can mention Malpighian body. So remember that they are mentioning these two things together, glomerulus and Bowman's capsule together. So the far away or the far situated tubule, the last tubule, that is the distal convoluted tubule. Now, if you see over here that there is this straight tubule that is there, right? This straight tube-like structure is called as the collecting duct. One more thing you notice that the distal convoluted tubule gets directly connected to the collecting duct. Other than that, you see these parts. It means that it's the extension for several other nephrons. Okay. So remember that the DCTs of many nephrons, they open into a straight tube which is called as collecting duct, many of which converge. Converge means they combine and then they finally open into the renal pelvis through the medullary pyramid, meaning this structure was there. So from there it will go into this part. If this is there, the renal, this part. So it will open over here. Clear? Understood, students? Any doubts?
No, no.
Okay. Now see, out of all of these things that people have studied up until now, the Malpighian corpuscle, okay, Malpighian body, Malpighian corpuscle, meaning Bowman's capsule and glomerulus together, the PCT and the DCT of the nephron. Where are they situated? They are situated in the cortical region, meaning they are situated in the cortex of the kidney. Cortex is peripheral or central, students? What did I tell you? Cortex is peripheral or central? Right now only I told you. What is it? Cortex is peripheral and medulla is in the middle. Okay. So the Malpighian corpuscle, PCT and DCT of the nephron, they are situated in the cortical region of the kidney, whereas the loop of Henle, it dips into the medulla, meaning like the other part is present in the cortical region, but the Henle's loop, that hairpin structure, it dips into the medulla. But depending upon how much it is dipping into the medulla, the whole entire nephrons, they are of two types. In majority of the nephrons, the loop of Henle is too short. It's not very long and it will only dip a little bit into the medulla. So you call those nephrons as cortical nephrons. Why? Because they are mostly situated in the cortex region. But those loop of Henle, like the loop of Henle of those neurons, which are very long, so they are going to run deep into the medulla. So now you can't call them as cortical nephron. Instead, you're going to call them as the juxtamedullary nephron. So nephrons, they are of two types in totality. The first type is cortical. Second is juxtamedullary nephron. Okay. Now, the efferent arteriole. Now, if you see over here, on all the, like on this yellow portions, you see the red portions coming through it, forming a network on top of it. Do you see that? So this network of blood vessel, it is actually a network formed by the efferent arteriole. Okay, it's like a network and we call it as the peritubular capillaries. And the part of that peritubular capillaries that is running parallel to the Henle's loop, it is forming a U-shaped vasa recta. Okay, and this vasa recta is absent or highly reduced in the cortical nephron, meaning those nephrons whose loop of Henle is not very long, it's very short, so they don't have vasa recta. But the juxtamedullary nephron, they have enough vasa recta present. Is it clear, students? All right. Now you people do one thing. You draw this diagram. You draw this diagram very nicely, like very artistic manner. And then also the structure of the nephron. I'll just clean it up so that you people have a practice. Okay. Yeah. So now you can draw it nicely because right today we are
Mom,
not
what are
making
the
copy
chances
notes.
that this will come in the exam for drawing?
For drawing, yeah, there are chances. Maybe let's just say you got a 5 marks question. Now you can support your answer with this drawing. Like it will support your answer, make you score full marks. Getting my point? That is why, as a precautionary measure, I'm asking you to draw. Okay?
Okay.
Done, students? This diagram is done.
Yes, ma'am.
Aisha, you're also done?
Yeah, ma'am, just finishing touches.
Okay.
Mom, you can scroll down.
Done, students?
Yes, ma'am.
You're also done, Aisha?
Yeah.
All. So now this was the basic structure that people draw. And also tell me one thing, did you people understand the whole entire ultra structure of the nephron and the tubules and everything?
Yes, ma'am.
Oh. Now see this picture here, it tells you that afferent is the one that brings the blood to the glomerulus. Efferent is the one that takes away. Okay, always remember that. Now, the next thing we're going to see is the urine formation. So now when our blood gets filtered out, the end product that is formed is nothing but the urine, right? So the whole entire formation process, it involves three main things. Firstly, the first step is glomerular filtration. Second is the reabsorption, and the third is the secretion. So there are three steps to urine formation: glomerular filtration, reabsorption, and secretion. Now, each of these steps are going to take part in different places in the nephron. The first step of the urine formation is the filtration of blood. Where does it take place? So remember that the blood gets filtered in the glomerulus, and that is why we are calling this filtration as the glomerular filtration. Now, on an average, 1100 to 1200 milliliters of blood is filtered by the kidneys per minute, which constitutes roughly about 1/5 of the blood pumped out by each ventricle of the heart in a minute. So the glomerular blood capillary pressure, it causes the filtration of blood through three layers. Now see, the filtration happens through the three layers. Where is that picture? They removed it. Do you people have a picture of that unit in your books? Because it's not here. See, there is this unit wherein the first three layers are the endothelium of the glomerular blood vessels, meaning the blood vessels of the glomerulus. Their endothelium is the layer number 1. Epithelium of the Bowman's capsule, meaning outermost layer of the Bowman's capsule, is the layer number 2. And then like layer number 1, layer number 2, and in between them there is a basement material which is called as the normal cementing material. I don't know why they have removed the picture. I had it. I'll show you. One second, I'm showing you the picture of how that looks like. Okay, students, can you see the screen?
Yeah, ma'am.
Now look, in this picture, this is how the three layers that I talked to you about, this is how it actually looks like. So first, like I said, there will be the endothelium of the blood vessel, this part. The first part that is there. Then there will be the epithelium of the Bowman's capsule, which is going to be the second layer. And in between them, there will be this basement membrane, which is sort of like a cementing membrane or sort of the membrane that is joining them together. Now, if you see, there are these holes present. Okay, these are the filtration pores. Through them, the blood is going to get filtered, just like how you people might have strained the tea, how it gets strained up, right? All the particles, they get left behind, and then the filtered tea comes out, right? So same way here, there are cells, or you can say the tiny spaces, okay, and the epithelial cells of the Bowman's capsule, they, like the name of the cells is podocytes, and they are arranged in a very intricate manner, as in they are arranged very close to each other, and so as to leave minute spaces, that is called as the filtration slit or the slit hole. So what happens is that the blood gets filtered out through these membranes, and all the different components from the plasma, they pass on, like they get filtered out, except for one thing, that is protein. Proteins are really big in size, so they don't pass through those slit or the pores, and they come, like they do not go into the Bowman's capsule area. Rest all the parts they do. So therefore, because it is such minute or precise kind of filtration that is happening, that all the constituents of the plasma, that is getting filtered away. So we call it as ultrafiltration. Clear? Any doubts?
Protein just stays in the body, or how does that get filtered out?
Yeah, what is someone asking me any question?
The protein, how does it get filtered out then?
Huh? Proteins?
How does the protein get filtered out?
It doesn't, because we don't have to remove that away.
Oh, so it stays in the body.
Because proteins are necessary for us, na? We don't throw it out. All the waste products they get. Okay?
So all the proteins that we consume are just in the body.
Hmm. Now, every single minute, the blood gets filtered through the kidneys, right? And like once the filtration has been done, the thing that we are left with is called as the filtrate. So the amount of filtrate that is formed by the kidneys per minute, meaning every minute the amount of filtrate that is formed, is called as the glomerular filtration rate. Okay, meaning every minute how much filtrate is being formed, that is the glomerular filtration rate. And in a healthy individual, it is 125 milliliters per minute, that every minute 125 milliliter is going to be filtered out. That is 180 liters every single day. Clear? But now my question arises that if, let's just say, every single day, if 180 liters of blood gets passed on or gets filtered out, but do we produce 180 liters of urine every day? Had we been doing that, we'll never come out of the washroom, we'll sit there only throughout the day. Imagine. So what happens? Like the amount of urine produced every day is almost 1 to 2 liters, and not more than that, depending upon the water intake. So what does that signify? It signifies that no matter, even if the filtrate is close to 200 liters, 99% of it gets absorbed, reabsorbed back into the body, because our body cannot afford the loss of water. And the urine release ultimately is 1 liters to up to maximum 2 liters, and that's it. Okay, so our body has that mechanism to reabsorb the good things back. And that is what we call as reabsorption process. Okay, so that was the second step of urine formation. Now, see, the kidney, they have built-in mechanism for the regulation of glomerular filtration rate. The GFR, it gets regulated. Regulated as in if it is, you can say, reduced, or if it is increased. So our body has a mechanism to maintain that. So one such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. Now this is a special region. Okay, this is present. Now where, like how is this region formed? It is formed by the cellular modification in the DCT and the afferent arteriole at the point of their contact. So the point where the DCT and the afferent arteriole are contacting, at that point, this special modification or this, you can say, association is called as juxtaglomerular apparatus. Now let's just say if in any person, the glomerular filtration rate, if that is decreasing, what does it mean by if it is decreasing? If, let's just say, that amount of filtrate is not being formed every single day. So in that case, these JG cells, meaning juxtaglomerular cells, they'll get active. And as a result of being active, they will release renin, which can cause, or you can say, which can stimulate to increase the GFR and take it back to the normal level. Clear? So this is a body's natural mechanism. We don't have to tell our body to do so. It's just that the amount of GFR that is formed every day, that amount will ask it to do so. Clear, students? Any doubts?
No, ma'am.
All right. Now this unit you people draw. Okay. This glomerular membrane. Always remember it is made up of how many layers? It is made up of 3 layers, and each of these layers have a specific function to perform. You remember that podocytes are present, which are the cells of the epithelium, and they create pores. Yes, this is also very important. I don't know, NCRT used to have this diagram, but I think it has been removed now.